You see that the title of this session is Ag Biotech is more than genetic engineering. And implicit in that is the belief that to some people biotech is only in genetic engineering. And that, of course, is true. To most of the public, the word biotechnology has become synonymous with genetic engineering. But biotechnology has always been a whole bunch more than genetic engineering. If you look back centuries and you look at the methods, the techniques, the practices, as Bill Weldon said, that humans have used over the centuries that have allowed us to use living organisms, plants, animals, microbes, to use them to make products or to solve problems, and also the techniques and practices we've used to change them so that they suited our needs better in making those products and solving those problems. All of those old, centuries-old things are a form of biotechnology, okay? It's biotechnology that's informed by our understanding of things like reproductive biology of plants and animals, classical genetics, microbial metabolic pathways, but it's all biotechnology, okay, and it's centuries old. When I hear people talk about educating the public and helping people understand, I have to tell you that the single most useful thing that you can ever do in helping people understand today's biotechnology is help them understand the continuum of technologies that got us here. Really to help them understand the history of agriculture is all it is. You don't ever have to actually say DNA. Just help them understand that everything has been genetically modified. In the absence of that, they think all this is brand new, and so anything you say after that is, they're still anxious because they don't understand there's a history. Okay? Modern biotechnology, is what we've been doing for the last 30, 40 years or so, is also a lot more than genetic engineering. Okay? Today's biotechnology is a set, this cluster of remarkable technologies, only one of which is genetic engineering. Okay? Monoclonal antibodies, protein engineering, cell culture, bioprocess engineering, all of these technologies are a form of modern biotechnology. What distinguishes them from earlier biotechnologies is that we're using now, not just our knowledge of reproductive biology, classical genetics, we're using our understanding of cellular and molecular biology. Okay. So the modern biotechnologies utilize cells and molecules. And by utilizing the properties of life at that level, at the cellular and molecular level, we're able to tap into or to capitalize on some really astounding properties of life at that level. And all of these properties of life make our technologies a lot better. It's not because we're smart. It's because 4 billion years of evolution has given us some really great things to work with. Okay? That's a lot of engineering and getting rid of bad designs and getting better designs. Okay? One of the properties is that cells can reproduce. In and of itself, therefore, technologies that are based on biology or on biological organisms and their parts are inherently more renewable. And this should help us in our quest for sustainability. Another extraordinary property of life at the cellular and molecular level is its specificity, extraordinary specificity. Okay? Because of that specificity, what you see is that we're able to detect smaller and smaller and smaller amounts of substances with greater accuracy. Okay, and we're able to get the results a lot quicker. So this means, because of that, we're able to diagnose diseases in plants and animals much earlier than we could before, okay? So we can start treatment earlier. We're able to also identify food safety issues, microbial contamination at much lower levels of microbes than we ever could before because of the specificity of biological molecules. Also, the specificity means anything we do to try to treat a problem, to try to solve a problem or fix it, is going to be more specific. It's going to be more precise. It's going to have fewer unintended effects. Whether you're talking about killing a cancer cell and leaving a healthy cell intact, or whether you're talking about putting a gene in a crop that will only kill the insect that feeds on the crop. That's more targeted, more specific, fewer side effects. Okay. Also, because of the specificity, any of the genetic changes we make now are more precise than ever before. Okay. Before we were mixing whole genomes, now we're mixing or adding single genes to genomes that we already know really well. Or now no longer just single genes, we're stacking them. Okay. To a biologist, probably the most stunning or awe-inspiring aspect of life at the cellular and molecular level is its unity. Okay, its oneness. All biological organisms, plants, animals, microbes, same building block molecules. They use essentially the same cellular processes. Okay? Because of that, I'm sorry Sonny's not here. He's the guy from NIFA. He's the head of NIFA. He's not the guy from, he's the head of NIFA, which is responsible for funding a lot of biotech and ag research. Because of that unity of life, any $1 that's spent in funding research in biology, for example, to find a gene in corn, if you found it in corn, I guarantee you found it in sorghum, and who knows who else you found it in. Okay, so that $1 you spent on that one project ends up being leveraged or amplified across all of these biological systems that have that same gene doing essentially the same thing. Maybe even Johnson grass has it and you can figure out a way to kill it better. The point is, $1 in research on one organism is meaningful for many organisms in many systems. Also, because of that oneness, what you see is that traditional barriers between industries, or not barriers, the dividing lines between industrial sectors are getting really blurry. Okay? Because, for example, a gene that you find or a protein that you find that's important in cell division, that's critical to the pharmaceutical industry, okay? Because cancer is a problem of cell division. But it's also really important to the agricultural industry because yields are based on increasing mass or numbers of cells. Okay, so you see this mushing up of what used to be industrial sectors are no longer clean. But probably the most striking aspects of the flexibility, the unity, the oneness of life that we now have at our disposal is genetic engineering. Okay? This ability to take a gene in any organism and move it into a different organism. We can do that because they all speak the same language, essentially the same language. Okay? You give a bacterial gene to a plant, it knows what to do with it. Okay. So because of that, we have this extraordinary flexibility, this access to all of nature's diversity that we never, ever had before. So when you look at what's been done so far with all of our access to this genetic diversity, what you see is that we've done pretty much the things we've done in the past. Okay, we've modified organisms, plants at this point in time with genetic engineering is the only technology I'm talking about now, to increase yields or decrease input cost. That's a lot of what breeding has been about for however long we've been breeding intentionally. Okay. Recently, we've begun to have a few consumer traits that have to do with changing the fatty acid profile. in some of our oilseed crops. But mostly we've been doing the same things that we've done before. And we've been doing it with the same crops, primarily the commodity crops, that a lot of breeding has been dedicated towards changing. But now we're doing new things, and our first speaker is going to tell you about some of those. He's Dr. Sekhar Boddupalli. Did I do that okay? You don't know how long I practiced that, all the way down the drive from D.C. Boddupalli. He's the global leader for R&D. Is something wrong? Oh. If you're driving.
If you're driving a Dodge Journey grayish color from a Hertz rental, please see the front desk.
Dr. Boddupalli's at your car. I'm glad. But it gave me another opportunity to say your name.
That car is running in the parking lot.
Okay. Dr. Boddupalli is the global leader for R&D into consumer traits that are being developed by Monsanto's vegetable seed company. The rest of his bio is in your program.
Can you all hear me okay? Good, okay. Okay, thanks for that introduction, I appreciate it. And I just want to today's morning has been very educational and iterates a few points that are critical in the global agricultural context. What I captured from today's conversation in the morning, as well as what's well documented now in reports, is that there are some dramatic shifts in global agriculture. And we talked about shifts in wheat production that's now going more to the Black Sea in Europe, and shifts from open field agriculture to protected culture in glass houses for tomatoes, peppers, cucumbers, more controlled environment systems. Shifts from North America to Mexico for agricultural production, shifts in the context of more agricultural production to Eastern Europe, to Southern Europe, to Morocco, to Africa and other nations, and India and China significantly picking up in agriculture. China is actually second largest economy on the globe now. And China accounts for actually more than 60% of global soybean trade. India is definitely picking up momentum in agriculture. So what all this means is that small disruptions in supply lead to major errors in global reliance on agricultural production. So if you look at what Africa, India, China, how much there is an increase in demand for just food production, and there are key drivers in organization of the supply chain across these countries as well. I heard from a Rabobank analyst 2 weeks ago that every day there is a McDonald's added on the globe now. Every single day. And the quick service restaurants are expanding pretty rapidly, with the middle class economy growing in some of these countries. What this means is more need for protein, more need for poultry, more need for feed that goes into the supply chain, as well as more food. What this means is actually a tremendous opportunity for agricultural companies, to retailers, to traders, to distributors, and to organize the supply chain that should be more streamlined in the context of production and what we call as precision agriculture. So agriculture today is not just more about talking a tool like biotechnology, as in biotech traits or breeding, etc. Agriculture is more than what we talk as cool science tools. Agriculture is more than simply talking about introducing a gene into a plant or combining plants to get native traits moving, etc. It is a systems approach. Agriculture is becoming a systems approach. 20 years ago, I definitely couldn't imagine, or I think most of us did not imagine, that a single industry called the cell phones would impact 5 different industries. I don't even need to buy my cable TV anymore. I can just use my Apple phone to actually $1.99, I can get an app to get all my cable channels. Who thought about that? Who thought about that we can do all the emails? I mean, it's really integrating all these technologies into a systems approach. So we as a Monsanto company definitely are investing into that approach. A system approach can be defined as a combination of related parts organized into a complex whole. So when a grower is harvesting crop each year, it's not just putting the seed into the ground. It's about understanding what the weather pattern is going to be. It's about understanding what inputs are required. It's about understanding the input and the output in a more precise way so that the risk could be mitigated and the return on the investment is more predictable than ever before. So as we actually create these system-based products, there is seed, there are seed technologies, there is integration of IT, there is understanding the data as it comes out of the field, and therefore there is need for investing in IT systems. There is a need for getting an iPad or whatever you have to the farmer so that they can actually manage the data as they get out of the field. And this is doable today. It's not something that is rocket science. And we are actually, one can envisage or enable building this into place. So what's happening in the context of companies like Monsanto and other companies that are investing in agriculture is that we no longer talk about a biotech trait. A biotech trait is as good as the germplasm that it gets into. And therefore, companies started investing in this. What is the base on which I put on this trait? So we are converging the breeding and the biotechnology platforms to develop total packages of traits and specific chemical inputs that would go onto it, and the weather that would be required to have a consistent quality of the product. Entire tomato industry is moving into, mostly into protected culture, whether it is glass house or plastic houses or why? Because they want to manage and mitigate the risk associated with stress, environment, pests, all other kinds of things. So that's the industry that's growing tremendously in Europe. Thousands of hectares of plastic sheeted. In fact, if you land onto some of the places like Almeria in Spain, what you see is an ocean of plastic. That's nothing but growing tomatoes, cucumbers, and peppers, and other protected culture crops. So these agronomic solutions that we're talking about include chemistry, integrated farming system to deliver new on-farm systems for total improvement in the output of the crop and the quality of the crop, while utilizing the natural resources in a more informed way, which is quite important for conserving the resources. And hence the mantra for us is produce more, conserve more, improve lives. The produce more is about understanding the performance of the genetics and the performance of the genetics in the context of the background, in the context of the environment. Conserve more is actually integrated farming system, and improve lives, as we heard from our speakers on improving the lives of the farmers as it relates to the exposure to the chemicals and the health risks, etc., as well as getting food that is more tasty, more healthy, and more fun. Food as a celebration, the choices of the food. So these systems are helping us in the context of whether it is marker-assisted breeding technologies to be able to combine native alleles or native traits more effectively from the global germplasm that is available. If I want a sour tomato, well, if I go to southern India, some of the sourest tomatoes I could find. And you can get all kinds of varieties of germplasm which is now available as elite material to breed with. And that's fantastic opportunity. And we haven't even started exploring the global germplasm diversity in crops such as vegetables. Is there a need for a biotech for a vegetable? Well, first, we haven't even explored the germplasm that is available to bring the best of the world diversity. And we started doing that with collaborating with even academic collaborators. An example of a product we actually helped develop that was innovation from United Kingdom by Professor Richard Mythen, who actually identified heirloom broccoli varieties from Sicily that contained higher levels or ability to produce higher levels of beneficial sulforaphane in broccoli. Well, we actually licensed his technology, and using marker-assisted breeding approaches, able to develop a broccoli that tastes fantastic. And if you go to any Sam's Club today, you will find a Beneforte broccoli. This is one example of how these technologies could help tap into what is available naturally out there, even before thinking about biotechnology approaches. So we look at biotech as one tool, a hammer, and if you have a nail, you need a hammer. You have a screw, you need a screwdriver. So we don't need a hammer for a screw, and we don't need a screwdriver for a nail. So these are tools. And these tools and it requires enormous amount of data management, and that's where the IT systems come into play. And when we start breeding, we are now implementing technologies that wouldn't even require the breeder to go put the seed in the ground. And that is actually sustainable agriculture, because over the last 5 to 6 years, we were able to save land and resources to go plant the seed to evaluate it in the field, because we can predict, looking at the chipping technologies, what traits are already present in these populations to be able to better select for the breeder, to be able to better select for the farmer. And what does this all mean in the context of healthy food? Well, that's where the vegetables come into play. Corn and primarily is going into feed. And Monsanto has invested in vegetable seed companies over the last 6 years in both open field and protected culture crops. And if you ask anybody across the globe, there is a need to improve consumption of fruits and vegetables. They're already healthy. Nobody cares about whether vegetables are healthy or not. The key thing that was highlighted today morning by the speaker is its taste, its affordability, its cost, its nutrition. Any consumer research across the globe indicates that those 4 are the highest priorities for improving vegetable consumption. The key driver is taste, it's taste and taste. And if I take my row crops like corn and soybean, it's yield, yield, and yield. It's not health, it's not anything else. So if you combine the two, vegetables means actually marketable yield, not just yield. The marketable yield as it relates to how can I actually have the people eat more and enjoy the food. And 10, 15 years ago, the breeders didn't have the opportunity to select in parallel for yield as well as other traits such as color, quality, appearance, convenience, etc. They had no way to do that. It was a sequential, empirical approach of going year after year, trialing, and some crops you have to wait 2 years for cycle, like onions. And for some crops, it is 4 to 5 months. So you can imagine how complex it was to breed for these quality traits. What they could select was, well, how much fruit can I get out of a tomato plant? And so that has driven the yields. And in the process, there was no effective selection for nutrients and effective selection for taste and things of that nature. So what we are trying to do is with the toolkit that is available now, the breeders have the night goggles to see through how they can select for better sugars, for better acids, for better nutrients, and things of that nature, and make it more pleasurable and enjoyable for the tomatoes, the peppers, and the cucumbers that we all enjoy to eat. At least I do. So all of this means that we need to invest in understanding consumer studies, understanding the sensory appeal that drives a French consumer for a Charente versus a Spanish one for a different type of melon. And what is also important is to pay attention to the post-harvest methodologies that handle our produce. We may have the firmest tomatoes that are harvested, breaker stage, jumping on the back of the truck and without getting crushed, but have absolutely no taste, to the greenhouse tomatoes that are handled like babies and deliver that fantastic flavor, and consumers are paying for such quality product. It's not the question of whether the consumer will pay for it or not. We have actually assumed that the consumer doesn't pay for them, but given the choice, consumers are looking for more flavorful, more colorful, better nutritious vegetables. And that's what we are trying to focus on. And I don't need to bore you with all the products, and that's not the intent here. I'm just trying to highlight what toolkit is being applied from all the way from breeding to selection to understanding the levels of the nutrients and levels of the taste components to validating them with the consumer, and then working with the chain, working with the retailers, working with the food companies to bring these products more effectively to the marketplace. And there are a few products you may want to check out: Beneforte broccoli, Melarange melon, that comes from Central America during off-season, but tastes as good as a main season Western Shipper melon. And Evermild onion that is locally produced in the United States, that has much lower, that's full of flavor and fewer tears, done through regular breeding. So I can go on, and Frascata lettuce with better crispiness and delicious and nutritious, etc. So these are some of the innovations that are coming through the pipeline. And as I mentioned, it requires precision farming. It requires molecular tools for better selection as well as phenotyping, ensuring that the aroma, the volatiles, everything is there for enjoying the product. So all this requires not just Monsanto doing it or some other company doing it. It requires a collaborative effort with other industry. As I mentioned, Beneforte was actually discovered in the academics. It's not our discovery, but we have the ability to bring those innovations to the marketplace collaboratively with other industry partners. So this all means that it's a all hands on deck approach, integrating IT, integrating consumer. So a few examples of how we are playing into that space. I mean, right here in North Carolina, we have 2 research centers, one here in RTP and another down in North Carolina, Kannapolis Research Center. And we are open doors to innovation, and we are collaborating with actually industry partners on the campus as well as university partners. And we are, if there are anybody that's interested in IT solutions and any of the other approaches for integrated farming systems, absolutely, we are welcome to, collaborate. And also what we did was, we have actually donated cabbage germplasm to North Carolina State University to promote and share our technologies and share our germplasm to develop vegetable products that we are not focused on, but could be, commercialized by university system. And so that's another approach we can take. We can improve the bandwidth. And as you all know, probably saw it we have donated, we are sharing our transformation technologies, genome information with academia and other industry partners. So all of this means we have to work together, and we are open to shake hands and get it going. Thank you very much. Let's switch it up.
Thank you, Sekhar. So Sekhar was talking about, for those of you who aren't familiar with breeding and agriculture, who are more the policy people, about using marker-selected breeding to tap into or discover existing genetic variation, okay, that we can shape in specific ways. Much of that has already been done with corn, soy, our commodity crops. I'm sure at some point, at least I assume at some point, you'll look to genetic engineering perhaps to introduce traits that maybe there's no genetic diversity for in those crops, maybe. Now we're going to talk about something really different, which doesn't have anything to do with agricultural plants as food. It's plants that serve as manufacturing facilities for vaccines and diagnostics. What is special about some of the new technologies, I want to back up for a second and say, you know, when people talk about the biotechnology revolution, or you used to hear them talk about that, the real revolution occurred in the lab. That's where the biotechnology revolution took place, and that we were able to use those tools to discover more and more and more about cellular and molecular biology, those facets of biology that gave rise to modern biotechnology. Because of that increased understanding, we now have this whole new host of technologies, many of which I do not understand for the life of me. We're able to go in and not add a gene, but do single nucleotide changes. We're able to go in and genetically modify something in an intermediate stage, but none of that genetic modification shows up in the offspring. We're able to trigger innate mechanisms in plants, DNA repair mechanisms, their innate immune system. We're able to use regulatory factors to keep a corn plant simply being a corn plant under really horrible, difficult conditions. Okay, we don't give it anything new. Okay, we just allow it to continue to be corn. And one of the questions that comes up, of course, is how will these things be regulated? We seem to have been penalized. The more precise we've gotten, the more knowledge we have, the higher the regulatory barrier we've had to overcome. Anyway, so Mike Wanner's going to talk to you about one of these new, very cool technologies that's being used to change tobacco plants so that they can produce vaccines and diagnostics. This is something we've been talking about for a very long time.
Thank you, Adrianne. Yes, so for people around my age, is the microphone on? So people close to my age, the transition phrase comes from Monty Python, which is, and now for something completely different. So we've talked a lot about agriculture for food products. I work for a company called Medicago, and Medicago uses a cousin of the tobacco plant to produce virtually any kind of protein in the plant as our biofactory. So our headquarters is up in Quebec City, where we have a research and innovation center, as well as a facility there where we can produce pilot lots that are used for clinical trials. The natural next sequence in the company's evolution was to have a commercial scale facility. So about a year ago to this day, we moved into a 100,000 square foot facility in Research Triangle Park, which is capable of producing in excess of 10 million doses of vaccine in a month, and we've already gone through and proven that via an agreement with the Department of Defense, the DARPA agency. So North Carolina is a great place to do business, and Medicago is a great partner in the state of North Carolina. We've appreciated all the support from NC Biotech and NC Bio and NC, you name it. This is a great place to do business. So I'd like to talk about the cool intersection of agriculture and biotechnology. And I'll deviate a little bit from the title of the presentation. I've never been known to be a rule follower, as my mother often pointed out to me. But we do use transient expressions, so it's not quite recombinant technology. But we bring along a tobacco plant, a form of tobacco plant, as our biofactory. In our facility, we have a seeding room in the basement where we take a tobacco seed and we drop it into a small potlet. After germination, we bring it up into a 27,000 square foot greenhouse. It matures there, and after about 35 days, it's in a condition where we're ready to introduce the biotechnology side. So again, the whole way up to that point, we have a production system that is really trying to get all those tobacco plants, anywhere from 10 to 15,000 of them in a lot, into a condition that they're very similar in phenotype, and we introduce our technology by a step we call infiltration. So we take those plants and we invert them into a tank that contains agrobacterium. So again, by itself, agrobacterium is not biotechnology. So if you stroll around the North Carolina State campus here today, you could see on some trees a large bubble that is really an infection from this bacteria that I'm talking about, which is agrobacterium. It forms on a tree, and we actually use that agrobacterium with the technology to introduce a gene of interest. And we do that by taking a gene sequence that's very specific and inserting it via gene construct carrying with the agrobacterium. So when we take those tobacco plants and invert them into a tank of agrobacterium, we pull a vacuum and we've drawn that agro into the outer surface of the plant leaves. The natural form begins to develop of agrobacterium, but we're delivering a gene of interest that may carry, for example, a surface protein, as we do for influenza vaccine. So now we'll have a protein forming within the plant. It has no DNA in it, so it's safe. It doesn't replicate, but it does begin to bud and shoot out the surface proteins, the same as are found on a native influenza vaccine or native influenza virus. So that's the confluence of nature and biotechnology coming together in a facility. From there, we go through a process that's pretty standard in terms of digesting the leaves of the tobacco plant that releases what we call a virus-like particle into solution, and then we filter out and use various steps of chromatography to end up with a highly pure and efficacious product at the end of it. I wasn't allowed to use slides at all, so I hope that conveyed to you how that all comes together. But to me, that's the beauty of taking what nature's already doing and introducing technology that ends up with a product that we believe is very effective in the marketplace. So we're in clinical trials. We've completed a phase 2 clinical trial with H5N1. We've completed clinical trials with H1N1. So those are the swine and avian flus. And beyond that, if you have technology using agriculture, the only reason to continue with it is if you have an advantage. So what we look to is an advantage in several areas. I think the first thing to think of is cost, and I think ag biotechnology offers a very strong cost advantage. So our facility, for example, the footprint of it is a 100,000 square foot facility. Cost, including all the equipment in it, was $37 million. There's facilities making vaccine in the area that are in excess of $1 billion. And so if you think about the long-term opportunities there, if we roll technology out across the globe, it's a bit hard to imagine that one might build multiple $1 billion facilities in Africa or other parts of the developing world, but not so inconceivable that we couldn't put a $37 million facility in some of those countries and expand the use of that technology. So we have a low cost of our footprint. We also have a low cost of production. So the greenhouse is a very effective and efficient way to grow plants and to introduce the technology. We recirculate the fertilizer that's used in the facility. We recirculate the evaporative cooling solutions that are used in the facility. So all that does is deliver a lower cost of doing business and a lower cost per unit of product that we will produce. So that's cool. Low cost is important. Also important for us is speed. So if we think back to the 2009 H1N1 pandemic, the virus was isolated and gene sequenced in April. There was not product to the market until October of 2009. So that's a 6-month period where the virus is spreading in the community and product is unavailable in the marketplace. So a convergence came along with Medicago's capability and the U.S. government's desire to close that gap from 6 months down to something more manageable. And an agreement was reached where we, through a proof of concept project, showed that we could produce 10 million doses of H1N1 in 30 calendar days. So we completed that project this summer. So cost is important. Speed is important. If we're not vaccinating people during a pandemic spread for 6 months, we get into a second wave, so that needs to be addressed. And then I think lastly is benefits of technology. And so we want to look for something that is perhaps cross-reactive. Can we maybe, by injecting one vaccine, deal with several different viruses? So there is some early data as well that we're able to stimulate some cell-mediated immunity that might cause some protectivity to other viruses. So to me, that's the cool part of bringing technology together with nature, using a tobacco plant, not necessarily very obvious, and bringing forward specific genes of interest so that we can deliver something very effective into to the marketplace. I also wanted to address briefly the importance of collaboration. We've talked about that a good bit today, and certainly in our circumstance, that's been a very important part of our ability to move forward. So we had this important agreement with the Department of Defense. We also have a very substantial investment from Philip Morris. Philip Morris knows a good bit about tobacco, I think, as we all know. We also have agreement with Mitsubishi Tanabe, working on a specific product that we announced in March for rotavirus. And we have a collaboration with the top 10 pharma to develop a vaccine product from them. So we recognize that we can't do things on our own. It does require collaboration. It does require getting around and out into the community, and that's also from the buying side, where it's important to deal with the community here in North Carolina. We've done a lot of work with BTech. We've done some fermentations with them. There's a number of CROs in the area. Alexandria Real Estate was a key collaborator with us in the development of the facility. A number of suppliers. We get enzymes from the marketplace, is a very important part of ag biotechnology, storage facilities, et cetera. So I encourage all of us to continue working together and sharing our ideas. That doesn't mean we necessarily do a deal the next day, but certainly making a lot of contacts and understanding where these technologies can come together and where we can collaborate, and where we can collaborate in terms of education and training our workforce, which we have a very great system here, which does that very well, is important to the ongoing building and growth of biotechnology here in the area. So that's what I wanted to cover primarily to get kicked off here. And Adrianne, I trust you'll ask a little bit about future thinking so we can get into that before we close up the session. All right, well, thank you very much. Appreciate it.
Okay. Thanks, Mike. One of the changes that we've seen in agriculture over the past, I guess, 50 years or so that's been really important for farmers is diversification. Instead of just growing large-scale commodity crops, low cost, what you're seeing is more value-added, selection for value-added traits, so that farmers can serve identity-preserved marketplaces. And this is all market-driven, okay? It's things like blue corn and white corn, okay? I mean, those are the food uses that fit in Mike Weldon's gourmet, I guess, luxury category. There's also selection in traits for molecules that have industrial uses, okay? And as I said, most of this is market-driven. Increasingly, perhaps, maybe it will be driven by policy, regulatory policy. We're not sure about that. But Rob Paarlberg is going to talk to us about how it could be that genetic modification and the use of these technologies is going to further differentiate what we think of as large-scale commodity crops into narrower segments.
Great. Thank you, Adrianne. I am going to depart a bit from the original script of this panel and raise a specific concern about the threatened future of ag biotech applications intended for direct food use as opposed to animal feed use or industrial use. And to situate myself between the other two panelists, I would say it's no accident that new science in fruits and vegetables is relying primarily on marker-assisted selection and not transgenic techniques, while the most exciting new applications of transgenic techniques are now perhaps being found in industrial products rather than in food products. Several recent events suggest that we may now be moving toward a future in which GMO industrial crops, including some very traditional GMO industrial crops like cotton, will be widely used and widely grown, at least in regions beyond Europe or tropical Africa, where highly precautionary regulatory systems may make that difficult. Genetically engineered feed crops like yellow maize or soybeans will be widely consumed by animals, even in places like Europe where they will not be widely grown. But genetically engineered varieties of food crops like wheat or rice or potato or white maize or horticultural products or genetically engineered food animals, including fish, may not be grown or raised outside of confined research sites and may not be consumed at all. This would be a highly damaging outcome. But let me mention three events over the past 2 years that suggest there's a chance that we're moving in this direction. The first event was a decision made by the government of India in the spring of 2010 to place an indefinite moratorium on the commercial release of genetically engineered eggplant, BT brinjal. Even though this crop had been examined and approved by India's own official National Biosafety Committee, the GEAC. They studied it for 3 years. They found absolutely no problem with it, and yet commercial release was blocked. The Indian Environment Minister put a moratorium in place. He didn't point to any scientific evidence of any risks to human health or the environment from this particular crop. But he'd been intimidated by activist groups in India who had mobilized against the introduction of this technology in particular. A second event that I'll point to is a decision by the government of China in April 2011 to pull back from what had been an expected commercial release of genetically engineered rice. Genetically engineered rice had been given technical approval in China by their Biosafety Committee in 2009, but in April 2011, a senior Chinese Environment Ministry official announced that Prime Minister Wen Jiabao, who had previously had very good things to say about genetic engineering, now thought the country should be more cautious on GMOs. And several months later, an official from the Ministry of Agriculture, in an interview with an important weekly newspaper, said genetically engineered rice may not be commercialized in China for another 5 to 10 years. The third event is one that we've been talking about already here today, and that was the success of anti-GMO activists in the United States last spring to get a ballot issue, Prop 37, onto the California election ballot for next month. If this measure passes, and it currently looks like it will pass, food companies may soon face the difficult choice of either putting a stigmatizing label on all of their products that have any genetically engineered corn or any genetically engineered soy ingredients. I say stigmatizing because the same groups that have put this measure on the ballot previously poisoned the mind of the public about what genetic engineering might mean for food consumers. They'll either have to put stigmatizing labels on their products or reformulate their products away from GE ingredients. And I assume they would have to do that not for the California market alone, but nationwide. If they decide to reformulate, it could be the final decisive move away from the use of genetically engineered foods for direct human consumption in the United States. I say the final decisive move because this is actually a process that's been underway for some time. I haven't wanted to admit it to myself, but recall that food service companies in the United States back in 1999 decided that they didn't want to use genetically engineered potato products and stopped purchasing those products from farmers. And so farmers in the United States who had been cultivating genetically engineered potatoes successfully stopped doing so. This product disappeared from the market. Remember that the United States has never commercialized genetically engineered wheat or rice, in part because of anxieties about consumer resistance in export markets, but also in part because of anxieties about consumer resistance here in the United States. These are food crops, food staple crops. And remember that even in the area of corn, yes, genetically engineered corn is used in some processed food products like chips or cornflakes, but food companies have all along decided that they did not want genetically engineered popcorn on the market. They didn't want the whole kernels sitting there staring at the consumer. So food companies have long been anxious about consumer resistance to these products and might not be eager to put the label on the product if that becomes the law in the state of California. So the California ballot issue could finally drive food companies away from using genetically engineered corn and soy in processed food products as well. So the question is, what would that mean for we'll start with U.S. farmers, if food companies reformulated away from genetically engineered corn and soy? And here, actually, the short-term setback to U.S. agriculture might be surprisingly small, which isn't necessarily good news, because you'd like to have U.S. agriculture a little more firmly in the camp of resisting a move like this. But for corn producers in the United States, consider the fact that only 12% of U.S. corn now goes into direct food use. 40% goes for biofuels. The rest goes for animal feed. So shifting 12% of the crop back to conventional non-GE corn might not require too much of corn growers. For soy producers, 98% of U.S. soybean production currently goes for animal feed. Of course, soybean oil does go to food use, and so there would have to be a significant adjustment there if food companies wanted to reformulate away from oil from genetically engineered soybeans. But for soybean growers, maybe some of that oil would find its way into an industrial use, into food use, into fuel use, for example, as in Europe. Vegetable oil is a usable biofuel. U.S. farmers would survive. That's my message. U.S. farmers would survive, at least in the short run. But a victory for mandatory labeling in California next month would do serious harm to investments in agricultural biotechnology science at universities like NC State, for example. And most important, it will significantly reshape the world of commercial ag biotech companies. It will send a clear signal that applications of agricultural biotechnology for food crops versus industrial or feed crops will have a greatly diminished commercial value from this point forward. And investments in the development of these crops will possibly dry up. Of course, if this happens, the real losers will be the agricultural societies of the developing world, where food crops are the core of their agricultural production, and the product that will be in greatest demand over the next several decades as the world goes from 7 billion people up to 9.5 billion people. Developing countries will need to produce much greater quantities of food over these coming decades. These foods will have to have improved nutrient content. They'll have to be grown with less water, less fertilizer, fewer insecticide sprays, and applications of agricultural biotechnology are one of the most promising ways of accomplishing these goals. But if the U.S. moves toward mandatory labeling for GM foods, this will undercut the argument that's currently made by U.S. industry and by U.S. officials abroad that governments in developing countries don't need mandatory labeling systems of their own. We don't have it, you don't need it. Well, now that we're going to have it, they're going to say, well, if you've got it, it's got to be the best practice. Europe has it, you have it, we need it too. And once a developing country adopts mandatory labeling, they will find that the easiest way to operate such a system inside an economy where much of the food currently being sold is being sold unpackaged and unlabeled, they will find that the easiest way to operate a mandatory labeling system in such an economy is to introduce no GMO foods at all. After all, that is how the European Union operates its mandatory labeling and tracing system. Just remove anything that might have to be labeled or traced, and the system suddenly becomes manageable. So if mandatory labeling becomes a global best practice, China may never commercialize genetically engineered rice. India may never commercialize BT brinjal. The countries of Eastern and Southern Africa may never commercialize the drought-tolerant GM maize products that are now being developed with assistance from the Bill and Melinda Gates Foundation. Nutrient-rich golden rice may remain confined to field trials forever. For both farmers and consumers in the developing world, this would be, I think, a lamentable, even a disgraceful outcome. It would be an imposition of the rich tastes of California, I think unintentionally, but nonetheless an imposition of the rich tastes of voters in California on the world's poorest people. It reminds me, I'm a political scientist, of why the authors of the American Constitution created a representative democracy rather than a democracy that made laws through direct ballot issues shaped by the passions and emotions and the poor information base of ordinary voters. But as I say, we've been marginalizing ag biotech applications for direct food use for quite some time, even in the United States. So it shouldn't be an altogether surprising next step down that path. Let me stop there.
Let's open the floor for questions. How much more time do we have? Less than 10 minutes?
You're already behind. Do we have any questions from the audience? Come to the mic, please.
California's done this before to themselves. I mean, they passed this proposition, I forget the number, but that said any establishment that had any compound that was either produced or sold or distributed or warehoused that ever caused cancer at any level in any animal, you know, you could be sued and fined if you didn't have a sign warning it. They just responded by wallpapering the state of California with the labels. And you know, I was out there at the time, and you know, you go into the grocery store, and at the end of every aisle is a little poster that says, warning, you know, the state of California has determined this establishment sells products that are known to cause cancer and reproductive problems in animals, and that includes wine and beer and milk and, you know, everything. And people just ignore it. Do you think there's a chance that that would be the outcome here, that there'd just be so much over labeling that it becomes background noise and has no impact?
I don't know. I've asked people from food companies what they will do if this passes, and I can't get a single answer or a straight answer. I don't think they yet know what they're going to do. Their first step will probably be to run to Washington and complain to the FDA that they need
Federal preemption.
a single standard nationwide, not just one thing for California and one thing for the rest of the country. Then the lawyers would get together and decide whether to challenge the California measure from Washington, D.C., and it might be challenged, and it might never come to pass. Even that, though, wouldn't be much of a victory for Ag Biotech, because then you are pushing unlabeled GMO foods down the throats of people who said they have a right to know. And that becomes a platform for the next stage in the campaign of outrage against an unruly industry that is disregarding the will of the American people. It's a nightmare scenario for industry. I don't think the food companies, well, I don't know what the food companies will do, but you are presenting maybe one way out of it. Just blind everyone with so many labels, remind them they've been eating this for 17 years without incident, and hope that those who respond to the labels by paying a lot more for organically grown food will be a relatively small share of the market.
The problem with that is that might be against the federal law, because you can only mandate labeling based on safety issues. And so that's why Ron said federal preemption.
Mm-hmm.
That's what.
Yeah, it's sort of like, in some ways, it's sort of like a group arguing that I want every package of grapes and wine to have a label on it that says this was manufactured possibly using non-union labor. And, it's not it's a social desire, and there are constituencies that want to know, particularly the unions, but, you know.
Its own. In the labeling issue, so what if a hypothetical company created a lettuce or a spinach, baby spinach in California that has a drought-resistant gene in it, so it uses much less water, and the label says this spinach was produced using significantly less irrigation water than conventional spinach? What would the consumer say about a label like that?
If that's all the label said, they'd be attracted. But if it said this genetically engineered spinach...
Okay, let's say genetically engineered food, but the
Yeah.
benefit is it's using significantly less water than the conventional food. So
Oh,
how
I
would
agree.
the consumer...
Like genetically engineered corn in the United States
So how would the
is
consumers-
significantly more drought tolerant and uses less water than conventional corn.
It
Yeah.
seems like the assumption here is they're going to just reject genetic engineering. But what if the label also illustrates or states the benefit of that genetic engineering? Will the consumer still reject that?
So we did consumer research in Europe as well as here, and consumer research indicates that less pesticide usage is something that resonates with the consumer. So taste, nutrition, convenience, less pesticide usage. That's consumer research. I'm not commenting on the label part. Consumers are interested in products that have less usage of pesticides.
You know, we haven't had mandatory labeling, but we've had essentially de facto labeling on milk for several years now in terms of BST.
Mm-hmm.
And people pay attention to that label, but they rarely pay attention to cheese and other milk products. So there we really have a real mixed bag of what the consumer sees and doesn't see. Mm-hmm. And so they can buy whatever they want. And in fact, they do, and most of that's driven by price.
The question is, where do you stop? I mean, even if
Yeah,
some
where
of
do
the commonly used ingredients are used in microbial fermentation, which is
Right.
genetically modified microorganisms.
Yeah, you're making
And
cheese
even cheese
with
is produced
genetically
using-
modified
yeah.
chymosin, right?
Yeah.
I would just guess that when the biotech industry has looked at this, it's not an accident that they've decided to throw millions of dollars into a campaign to defeat the measure.
Sure.
There's the biotech industry and the food industry, and those actually are two different things. The industry is not uniform.
Question over here on your stage left to you all. I raised the CalProp 37 question earlier because I do see that this is really potentially very profound impact. And my follow-up question to this panel is, since there is so much evidence of safety, and since the putative concerns that are driving these consumer right-to-know issues are about safety, why not get ahead of the issue in a proactive way by voluntarily empowering some kind of third-party, non-aligned underwriter's laboratory type seal that communicates, yes, indeed, these ingredients are in these products, and they've been tested and proved to be safe by international standards. And instead of that, while that would incur costs to the producers, it would probably incur a lot less costs than spending millions and millions of dollars fighting consumer protection lawsuits, which was the primary impact of Prop 65 that the other person referred to. It just has generated a boon for trial attorneys, but it hasn't actually protected consumers one bit. So there's an opportunity to turn a negative into a real positive, I think. And so what do you think of that type of proactive, transparency-driven approach?
In the European Union, for the last 10 years, every national science academy, the French Academy of Science and Medicine, the Royal Society in the UK, the German Academy of Science and Humanities, and the research director to the European Union itself, they've said repeatedly in writing, on the basis of publicly funded research, not industry research, we find no evidence of any new risks to human health or the environment from any of the genetically engineered products that have ever been commercialized anywhere. The EU says all this shows that genetic engineering is not per se any more risky than conventional plant breeding. The impact that has on the attitude of consumers in Europe is, as best I can tell, zero. Consumer attitudes, they're remarkably insensitive to risk. As was said earlier, if you get I think Jack Bobo said it, if you get a benefit from something, you push all the anxieties about risk to the side. The problem with genetically engineered foods so far for consumers in rich countries is they don't provide any significant direct benefit to consumers. They provide enormous benefits to growers. They provide general environmental benefits. They provide benefits to seed companies and technology innovators and to scientists. But the final consumer doesn't get a product that tastes any better or looks any better or smells any better or delivers any more nutrition than the conventional alternative. And so even imagined risks are enough to make that consumer resist the introduction of the new product. So I wish it were simply a matter of informing consumers about the scientific consensus that there's no risk here, but the absence of risk is not the same thing as a presence of a tangible benefit.
One more question, because as you know, we've been told we've already over, so you're the last guy.
Since these goods that would be regulated by Proposition 37 are in interstate commerce, wouldn't it be subject to challenge as a unconstitutional state regulation of interstate commerce? The classical law school case is, a state cannot regulate the length of a freight train coming through its state. And it's the same sort of thing. These goods have to be shipped to 50 states in the country, and you can't have states imposing separate regulations on those goods. That's the same principle of free trade and free movement of goods in the European Union. And what they wanted to get away from was individual nations from regulating it, and it's based on the United States Interstate Commerce Clause.
I think that's one of the angles that people are looking, one of the legal angles people are looking at.
I would think it'd be the first challenge right out of the box.
But probably the most important message is, okay, so let's say in California, it's solved. What about other states? So this will keep cropping up, and probably the most important thing is if we win, we still lose. Because as someone said, it looks like we have something to hide. And that's an odd place to be in.